We are standing behind us there is our root rot disease in nursery, which is primarily what I think Jamie asked me to talk about today. The problem with our root rot disease nursery is that we often don't see disease until a little bit later in the season. And particularly this year when it's been such a dry year, we're not going to see root rots pop up until a little bit later. We have already put irrigation on this field. I think you said about 1 inch last week. And it's already pretty dry, so we will see what kind of season we end up for root rots. So I thought what I would start with talking about is give some background on root rots, have a few demos, and then I'll talk about some of the other diseases that we work on. And then if there's still time, we can just kind of wander through the disease nursery. We're doing some interesting things with evaluating different germplasm lines, and you can start to see right now just the different growth habits that are out there. So we'll wander through, but the plants are, I think Christine, yeah, our plants are about this big right now. We just pulled some up. Only I think kind of at the 4 above ground node stage, so really too early to start seeing anything. Hector, can we find it again? Like, oh, keep that one for Hector. There's something feeding on the roots, but I don't know what it is, so you can look at it. And these are full of pea leaf weevils as well. So I can pass them around if everyone wants to see pea leaf weevil damage. They are starting to nodulate as well, so that's good. And what we do with our disease nursery is we basically do everything you're not supposed to do to grow peas. And the number one thing is we really push the rotation. So we just rotate this field kind of back and forth. We do peas one side, they do wheat the other side, and then we're always just going back and forth. So we have peas every 2 years in this site. We also over irrigate. We push a lot of irrigation on the site, particularly into July when we start seeing the root rots develop. And that really helps push the root rot. And we actually started out, when we first started setting up this disease nursery about 6 years ago, we actually put infested grain spread out onto the field, and that's where the Fusarium colonizes that grain. So for the most part, we're pretty successful at getting Fusarium root rot. What we are not successful at getting at all is aphanomyces root rot. We've been trying to establish disease nurseries for this pathogen for the number of years, and it seems like no matter the fact that we're doing everything wrong for peas, we just can't get aphanomyces to establish. And really we're putting peas in every year on the site where we're trying to get it to go, and we haven't seen it yet. But keeping our fingers crossed. Anyway, I can pass these around if people want to have a look at healthy peas. And what I want to start out with then is really just going with a history of root rots, the 2 different types of root rots. I'll show you some examples. And then we'll move into some of the other diseases. So what I did was printed out some pictures of kind of a field, different fields when you see the progressive stages of root rot. So pretty early on, kind of the first clue is that you will start to see little yellowing patches in your field, particularly in areas that are low spots. Water along here, you can see it's along water tracks. There's kind of a hill here, hill here, and you can see that you get these yellowing patches. So people will see this in their field and think, oh, okay, just a little yellow patches, no big deal. And then they will plant peas again, and I can pass these around, people can look at. They will plant peas again, peas or lentils again 2 to 3 years later, and then all of a sudden you'll see that these little yellowing patches are starting to spread much more rapidly across the field. Then we often see them kind of move with your seeding equipment or combine equipment. You can see them kind of move up and down on those tracks through the field. And then in a really bad year, and we don't see this all the time, but then you can see how those yellow patches that maybe you had 4 or 5 years ago, or suddenly the entire field is completely yellow. And you can see in the background here, there's a couple fields that we really weren't be able to harvest at all. So I think those are some nice photos that show that progression of kind of, oh, there's maybe something wrong with my peas, a few little yellow spots, a couple of rotations of peas or lentils later, and you see a complete kind of failure of your crop. Okay. And we have 2 kind of main organisms. The one that gets all the attention and really causes the most damage is Aphanomyces eutyches, and there's a picture of it here. And I also have some plants to pass around. Hopefully everything doesn't blow away. So this is what Aphanomyces looks like, and just showing you in comparison to kind of a healthy plant. This is the extent of the damage that you can see. Really a big difference. And if we let these grow for another week or two, these would probably be completely dead. And what?
Dead plants survive, Syama?
What's that?
Will dead plants survive?
These plants?
Yeah.
Probably not. And this is like worst case scenario in the field. In the greenhouse, where we're really giving them a lot of water. We've seen plants in the field that surprisingly have survived and given a good yield, even though they can look pretty bad. And I have a picture of what that can sometimes look like. But in the greenhouse, these guys, they won't survive. Yeah. And I can pass these around too if everyone wants to take a closer look. Yeah, and then this is what, I guess why we have it out. The other pathogen that we're dealing with is Fusarium Root Rot. And you can see that there's really a big difference in symptomology in a perfect world. With Fusarium, you get this kind of blackening of the taproot. We usually see it start at the point of seed attachment and spread from there. And then you'll start to see that the lateral roots get brown, but you don't get that same loss of root material like we did with Aphanomyces root rot. And so what we have here in our disease nursery is actually we're evaluating for Fusarium because we can inoculate the field, and like I said, for Aphanomyces we have a really hard time getting it to establish in the field. So I will pass these around again. What else? The pictures I have. More pictures.
You all get black like that, Syama?
Fusarium gets black like that, yeah. And what often happens, I think I have a picture here. What often happens in the field is we get a combination of both Aphanomyces and Fusarium. So you can pull out the roots and they look really black, and then it's hard to tell, do I have a Fusarium problem or do I have an Aphanomyces problem? It's really hard for me to tell you right away what you have if you kind of pulled out your roots just looking like that. Because this blackness would say it's probably Fusarium, but then you look at what was maybe left behind and there was likely Aphanomyces that came in first, and then Fusarium comes in on top of it. So we've done some studies looking at changes in the pathogen levels over a growing season, and we do see that Aphanomyces kind of peaks mid to late June. Is typically when we get a lot of the rain, and then we see Fusarium come in after, usually in July when things have dried up. So for the most part, we're talking about a pathogen complex where these two pathogens have very different environments that they like to survive on, but they're taking advantage of the fact that both of them can infect. So we can pass this around. But what we can see sometimes, might have lost that. One of my pictures blew away in the wind, and that might have been one of them. Yeah, I think I lost that one. What we can see sometimes, though, is that despite that damage that you see on the roots, and particularly with Fusarium, and in our disease nursery we see this often, we can have like this much of the root left and it'll look black and really ugly, and we can pull off a really nice yield off of that crop still. And usually in our disease nurseries and some of our field trials, we're still kind of pulling off that 3,000 pounds per acre yield even when we have severe root rot. So we're still trying to figure out what that relationship is between root rot and yield. We've done 4 years of field trials, and I've tried to build some regression lines and relationships, and it's just it's flat. Sometimes you have so much disease and you get very little yield, and sometimes you get just as much yield. So we can't quite figure out what it is that's going on with that. But and the big problem is that it's hard for us to do field trials where we could compare a healthy check to our disease checks. And then just, I guess, for interest's sake on our pathogens, this is what they look like. I think those that have been following the root rot story know that Aphanomyces is a fairly recent player on the prairies. We didn't think we had it for a number of years, and then 2012, 2013 was kind of our first report of this pathogen. And part of the reason for that is when we look at the cultures, it gives us a clue for why the problem is. So Aphanomyces growing in culture looks like this. It's nondescript, very flat growing, doesn't really jump out at you on a culture. When we look at Fusarium, it looks like this. So nice, hot pink color, very, very distinctive. It tends to grow a lot faster than aphanomyces. So when we were trying to figure out what the root rot problem was and putting these diseased roots out onto plates, this is what you think. This is what you see first automatically, right? And so it was like, oh, well, it's just a Fusarium root rot problem. It's not until we went to doing DNA diagnostics of roots that we could pick up that aphanomyces was very prevalent in most of our soils. And then we have a couple of different, another challenge is that we do have a couple of different Fusarium species as well that cause disease. So this one is Fusarium Avenacium, kind of a very common generalist pathogen that can really infect everything a little bit, but it tends to like peas the most. So you'll find it a little bit on cereals, you'll find it a little bit on canolas. You plant your peas and it's like, oh, that's my favorite food, and really infects peas. And then the other one that we have is Fusarium redolens. This one is a pretty minor pathogen, seems to like the pulse crops, doesn't cause a lot of disease on its own, but you put it together with aphanomyces and it just goes crazy. So there's all sorts of interactions that are occurring in the soil between all these different kinds of pathogens that really drive the disease. And then there's another Fusarium, Fusarium solani, that is actually what we primarily have here in our disease nursery, but we're not allowed to culture it because it's been reclassified as a risk group 2 organism. Even though it's everywhere in the soil, we still can't grow it in the lab, so I don't have a culture to show you. But anyway, those are kind of our 3 major culprits there, and I can pass those around. Everyone wants to look. Yeah. Well, I think just again to kind of drive home the difference between aphanomyces, I have a little kind of disease cycle here for aphanomyces, and really what the message here is that aphanomyces is dependent on rainfall. So it produces these long-lived resting spores in the soil that get shed behind from the infected root that's going around. You can see that brown color is just full of these oospores. They get shed behind in the soil after you harvest your peas, and they will sit there in the soil, particularly in these dry conditions. They'll just sit there quite happily until you add peas back into the mix, and then you add water. So the recipe for aphanomyces is peas or lentils plus water. And then the oospores germinate. They produce this structure down here. It's called a zoospore, and those are actually little swimming spores. So if there's enough water in the soil and those soil pores open up, they can move towards the root, find the root, infect it, and then you get you complete the disease cycle where you get that pathogen left behind in the soil again. So one of the things that we're trying to figure out is, well, we're in a dry cycle now. When we first found aphanomyces, we were in the middle of a very wet cycle. Like I think from about 2010 to 2016, it was very unusually wet. Now we seem to be moving into a dry cycle. So, fingers crossed, we will maybe see this aphanomyces problem decrease. We've certainly seen that for lentils. Lentils seem to get less disease in a dry year, whereas peas, we're still trying to kind of tease that out a little bit. What we see is that if there's just enough moisture, the peas will still get infected, and then they're subject to a very dry condition, and then they're kind of get hit with a double whammy because they've lost their roots kind of look like this, and then it's really dry, so they can't deal with the dry conditions. Whereas lentils, we haven't seen that to an extent, but we are still trying to figure out what that kind of interaction is between rainfall and dry soil. And then finally, just for Fusarium, because I always say aphanomyces gets all the attention, it is kind of our A-list pathogen, but in all of our fields that we've surveyed that had root rot, about 50% of them were positive for aphanomyces, the other 50% positive for Fusarium. So really we're dealing with a pathogen complex and 2 different diseases, a Fusarium root rot and aphanomyces root rot. So Fusarium can cause damage in its own right. I don't know where that picture went, but it does tend to not cause as much yield loss. And so I think that's why it gets a little less attention. But for Fusarium, like I said, what happens is it starts at that point of the seed attachment. You'll see little brown lesions start on the roots, and I was hoping that we would see some today from the disease nursery, but it hasn't started yet. That gets progressively spreads through the taproot. And really, if you want to know if you have Fusarium, the thing to do is to usually slice open the taproot, and you'll see this really bright red staining in the vascular bundle. And that's a really good indication that you have Fusarium. Unfortunately, you don't see it 100% of the time. So if you don't see it, it doesn't mean you don't have Fusarium. But if you do see it, then you know for sure that you're dealing with a Fusarium root rot issue. And I think that was all that I had to say about root rots. Is there any questions before I move on?
Yeah. How long does that take to rotate out a piece of lentils before it would be okay?
Yes, it can take a long time. We're saying anywhere from 6 to 8 years rotation out of peas and lentils. One of the questions is if we're in this dry cycle, can that rotation time be decreased? And I should mention my field book. So we are actually doing a number of kind of agronomic trials looking at some of these questions around rotation. So this is my field book put together by my lovely technician Christine over here. And basically every single one of these tabs is all the different field trials that we're doing, all revolving around root rot. So we have one that's looking at cover cropping, I think that Yvonne talked about this morning. So we are looking at whether the use of brassica cover crops. There's been a lot of reports in the literature that brassica cover crops will help break down those oospores in the soil. So we're looking at the use of brassica cover crops. Then we have our disease nursery. Then we have a rotation trial. So what we're looking at that is that question of how long do these oospores survive in the soil. Just kind of with a regular rotation with wheat and canola and changing the frequency of a pea crop. And then on top of that, we're also looking at, well, what if we replace a pea crop with a non-host pulse crop? So something like fava beans, chickpeas, or soybean, how does that affect the inoculum levels that we're seeing in the soil? Just to make sure, our messaging has been, you can plant these crops, we want to ensure that they are not actually contributing further to the problem. And then we're looking at our PRRs, our seed treatments. So we're continuously evaluating different seed treatments that are coming out from different companies that are coming out in the pipeline, and that includes both chemical and biological treatments. And then we have an intercropping trial, again, where we're looking at whether intercropping with some of these brassica species that maybe are producing some of these biofumigants will reduce root rot severity. Just 1 year of data from last year would indicate that maybe they are not effective at reducing root rot, but we did actually see a yield bump in that intercrop compared to the monocrop. And so it could be an option if you have some root rot starting in your field, that if you intercrop, you can still pull a better yield off of it. Then we're looking at this one's more seed treatment products, and then we continue to do our surveys. So we're trying to just kind of tag and catalog what all the diseases are, and then we are looking at germplasm screening, which is one of the trials that we have in here. So this book is basically everything that we're doing with pea root rot, and then there's like one tag here for bacterial blight and one tag for fava bean. So a little bit of work that we do on other trials.
Are any of the breeding programs specifically trying to breed resistance to?
Yes. So, and that's part of what we're doing here. There is not a lot of germplasm available that has documented resistance to aphanomyces, but there has been some released by USDA and the group in France. And so the Crop Development Center up at the University of Saskatchewan is working on breeding, and we're also starting a breeding program here in conjunction with our breeder up in Lacombe, with DJ Bing. So what we have out here is some of those germplasm lines. Because anything down the road, anything that gets released, I think is going to need to have resistance or partial resistance to both aphanomyces and Fusarium. So we're doing the assessment for Fusarium here in our disease nursery, and then they do the assessment for aphanomyces up in, at the University of Saskatchewan. And the idea is at some point we will kind of pyramid those two resistance, if we can find them. But, yeah. Any other questions on root rot?
briefly comment on your biological seed treatment? Because we all know how fungicides work per se, but what's actually in the pipe with respect to biology?
Yes, we are trying to evaluate some products that are already on the market, but not necessarily registered for pea root rot, with the idea that if there's anything that works, if it's already been registered for something else, it'll be much easier to move a registration to peas. And what we're looking at is products like trichoderma, I think clonostasis is one of them. And these are beneficial fungi that have been shown to be very active colonizers of the roots, and with the idea that if you can get those fungi, those beneficial fungi colonizing the roots and kind of creating a protective boundary before you have your pathogen coming in, that maybe we will see some improvement. And then I know one of the scientists out at Swift Current was doing some work with mycorrhiza as well, with the same kind of ideas that if you get these defense responses activated, will that help to reduce pathogen infection? Yeah. Exactly. Yeah, exactly. Yeah. And we'll see if it worked. I did all my grad work was on biocontrol of Pythium root rots, and they worked, but you know. It's such a different setting in a field situation where you need to get the colonizing active very, very quickly. But yeah. Okay. I don't know how much more time I have. If I have time to talk quickly about beans. Yeah, I think I'm good for time. Okay, so I am like the root rot queen, is what they call me. Actually, my nickname is Beyonce, but I can't just study peas because we do grow other pulse crops here in Alberta. So I am going to talk very briefly about some of the work that we do on dry bean and also on fava beans. And I don't know, are any of you here dry bean growers? Well, there's a couple of you. Oh, yeah. And how many of you are canola growers? Probably a lot more. Because one of our big problems on dry bean is sclerotinia, which causes white mold on dry bean, but it's the same pathogen that causes sclerotinia stem rot on canola. So we do a lot of work on dry bean, and I think we're just starting to work with Charles a little bit, maybe under Kelly Turkington's umbrella, looking at some sclerotinia of canola. But dry beans really are the most susceptible to white mold out of all the crops out there that sclerotinia can infect. Dry beans really seem to do the worst. And so what we do is we work very actively with the breeding program here to try to build resistance, try to build some sort of partial resistance into beans. And so we screen all of the material that comes out of the dry bean breeding program for white mold. And we do this both in the field and in the greenhouse. In the greenhouse, what we do is we take a plug of the sclerotinia pathogen here, and we cut off a stem, and then we just kind of plunk it in there. And then you can see, I think this one was kind of the advanced infection. And this one is actually a line that has some fairly good resistance to white mold. And you can see that it moves down the stem, but it kind of stops when it meets a node. And that's good, that's what we want to see. And then I think there's another one here where we can see that the white mold has kind of moved down the stem, and then it moves past the first node, and it keeps on moving down. And then eventually we'll see that the whole plant dies from it. And I can pass this around. I think there was little, some of the sclerotia was starting to form in some of them, if you guys want to have a look. And what I mean by sclerotia, I'm sure everyone has heard this by now, is familiar with sclerotia. So these are the little guys that get left behind after you get infection, and it's the same in canola. It's an interesting pathogen because the sclerotia that gets left behind tends to take the shape of its host. So on bean sclerotia, you'll see is quite large. Some of them actually look a little bit like bean seeds, whereas the sclerotia that comes from canola is usually quite small. It comes from the stems, and so they look a little more like little mouse turds or something, whereas these ones can be quite large. So this gets left behind in the soil. It just kind of looks like rocks. Again, it can sit in the soil for 2 to 3 years, and then once the conditions are right, you get a lot of humidity, a lot of moisture in the soil. It germinates to produce these little mushroom-like structures that then get released into the air and cause disease. And I have a grad student behind me there, John Reich, who is looking at a project to be able to predict white mold and sclerotinia in both canola and dry bean by looking at detection of these spores that get released into the air. If we can determine when they're flying, what the conditions are that they get released, then we can maybe tighten up our fungicide applications and know when the best time is to apply fungicides. So that's another, I say a small part of my program, but a big part of John's PhD. That's a pretty big project. So I can pass these around too. And then lastly, before we kind of just take a little walk through the disease nursery, I'll talk quickly about fava beans. Although, are there any fava bean growers in the crowd? Oh, we have one back there. So we're doing a little bit of work on fava beans. Really, it's a fairly new crop to Alberta, so we are trying to determine what are going to be the pathogen problems. And kind of our clue from what occurs around the world is that chocolate spot, which is caused by a disease called botrytis, is really going to be our big problem. So we've been out looking for botrytis and testing it on a lot of our different, or testing it under a lot of different conditions to see how it likes to infect. And this might be very, very sad looking fava bean plant, but you can see how devastating chocolate spot can be once it gets going. So we have here kind of an early stage infection where you just see little spots on the plant under the right conditions. So it goes from what we call kind of the passive phase to the aggressive phase. And then that's when the pathogen starts to cycle through, and then you can get complete defoliation of your plant. So I can pass that around. And what our pathogen is here is it's botrytis. It's actually a cousin to sclerotinia, so very similar conditions to get botrytis as sclerotinia. It also produces sclerotia, much like sclerotinia does, but it also produces these masses of airborne conidia, and that's what the infective stage is, and that's why you can kind of move to that aggressive phase when you just get this continuous cycling of these conidia being formed under moist conditions. So I can pass that around too. And I think that was all I had to say about dry beans and fava beans, if anyone has any questions about that.
Chairman, you like bad looking stuff.
I know, I do like it. Yeah, the uglier it is, the better. Okay, so what we'll can try to do, like I said, there's not much to see in the disease nursery now, but we'll try to take a walk through and just kind of look at what some of those different germplasm lines can look like. So unfortunately, they kind of ended up in the portion of our field. Christine's back there. We had I think it's a really nice demo actually. We had a missed pass putting edge down on the field. So unfortunately, our germplasm line is kind of in that missed passive edge. So you'll see that it's quite weedy, but it's a good demo to show that edge really works and don't miss a pass when you're applying it. But so we can walk through here just quickly if people are interested to see what they look like. I think we're right down here in our weedy spot. So our first two plots here are actually the results of crosses that we did between susceptible adapted cultivars, so Chrome and Comfort, which are kind of elite pea cultivars. So we crossed those with some of our resistant germplasm. We end up with a handful of seeds. And what we wanted to do is take those out into the field right away and see if we actually manage to transfer any resistance from our resistant germplasm lines into our kind of elite adapted cultivars. So we're growing them out in our disease nursery. and we will see what they look like. If you come and have a closer look at them, you can see right away that they do have a little bit of a different growth habit. And that's because unfortunately, a lot of our germplasm lines that have resistance are still kind of the fully leafed, indeterminate, viney pea types. So it's going to take some work to try to transfer that resistance to adapted lines. So if you look down here, you can see already that they're fully leafed. Some of them, I don't know if we're in the right spot here, some of them come from a colored, the purple flowered pea, and so you can see right away that some of that redness is coming out onto the stems. But I thought it was interesting that that phenotype does carry through. And there is some evidence to suggest that resistance might be linked to flower color. So those purple flower colored varieties might have better resistance, and it might be hard to break that linkage to get resistance into our adapted peas.
So don't you treat for the weevil?
We did not treat for the weevils because we haven't had a big weevil problem at the site before, and so we thought we would be okay. And there is a lot of weevils here. I don't know where. Yes, so I don't know where Hector is, but this is a good. Oh, right here. This is a. I don't know if he wants to say something.
He wasn't looking.
He's counting notches. Yes. Yeah.
See, and it doesn't, you got to put it on the seed.
Yes, exactly. Yeah, you got to do a seed treatment, and we don't seed treat these with fungicide because we want to get the disease, and so we just didn't even think to put on the insecticide instead. But
I see that. I know.
yeah, we did have a project with Hector looking at pea leaf weevil and root rot interactions, and I will never do that again because counting those pea leaf weevil notches, that is painful.
How effective is that, talking about those diseases, how effective is that in Tegozola? Does it do anything?
Yes, another really good question. Yes, we've done some field trials with it. Once you have really high disease pressure, we didn't see that it really reduces root rot or it doesn't increase yield. But at sites where, where I showed pictures where you're just starting to get those yellowing patches and disease pressure is pretty low, we did see that you get a benefit from it kind of at the lower disease pressure sites. So it does mean that you might have to have some knowledge or prior history of your crop and recall, okay, did I have a really bad root rot problem? Because then a seed treatment's probably not going to help you. But if you're just starting to see some patches, then you're a good bet to use the seed treatment product just to keep it down and stop it from
That's the only one, right?
Yeah. Like I said, we are evaluating other products, but it's not up to us whether they get registered or not. So these are our two plots that have our crosses, and then if we kind of move down the row here, these are all of the lines that are the germplasm lines that are meant to have some sort of resistance. You will see that some of them are growing quite nicely, and then you'll see that some of them are struggling a little bit more. And like I said, it's because these unfortunately are not lines that are really adapted for our environment at this right now. Yeah, I am. I'm a breeder now.
Nice, congratulations.
Yeah. I don't know when I get my breeder license. Yeah. But anyway, so they're still pretty small, and so you can't see what a lot of the differences are, but I think it's just kind of interesting to see them out in the field. Yeah. And I think, that's all I kind of have to say, unless anyone has questions. Did I stick to my time? Did I go over? Right on time.
I guess join me in thanking Syama then.
Thank you.